All right, these are the notes for Target 2C. So make sure you use this PowerPoint to take notes. Over this topic, it's going to be about the cell cycle. I'll read it to you after we watch our kind of introductory video for the cell cycle. So make sure you have a pen, pencil, paper, whatever you need so that you can use these notes for your tests and your assignments. All right, so for this target, we are going to start out with a video about a woman named Henrietta Lacks. She was a Black woman who had cancer and who some scientists experimented on her. Well, I guess not experimented, but they took some of her cells when she went into the doctor when she wasn't feeling well. And they took those cells and they experimented on those cells. And they did not tell her about that. They did not tell her family about that. And they made lots and lots of money from her cells and her research, or the research that they did on her cells. So it's kind of a controversial topic because most people think that she should have been paid or her family should have been paid for the research that they have done on her cells. And then now, today, doctors, scientists can't just take your cells, your body parts, and do experiments on them without your knowledge. But this is the video of Henrietta Lacks, which is going to lead us to the cell cycle, which is part of our Target 1C.
Imagine something small enough to float on a particle of dust that holds the keys to understanding cancer, virology, and genetics. Luckily for us, such a thing exists in the form of trillions upon trillions of human lab-grown cells called HeLa. Let's take a step back for a second. Scientists grow human cells in the lab to study how they function, understand how diseases develop, and test new treatments without endangering patients. To make sure that they can repeat these experiments over and over and compare the results with other scientists, they need huge populations of identical cells that can duplicate themselves faithfully for years. But until 1951, all human cell lines that researchers tried to grow had died after a few days. Then a Johns Hopkins scientist named George Guy received a sample of a strange-looking tumor, dark purple, shiny, jelly-like. This sample was special. Some of its cells just kept dividing and dividing and dividing. When individual cells died, generations of copies took their place and thrived. The result was an endless source of identical cells that's still around today, the very first immortal human cell line. Guy labeled it HeLa after the patient with the unusual tumor, Henrietta Lacks. Born on a tobacco farm in Virginia, she lived in Baltimore with her husband and 5 children. She died of aggressive cervical cancer a few months after her tumor's cells were harvested, and she never knew about them. So what's so special about the cells from Henrietta Lacks that lets them survive when other cell lines die? The short answer is, we don't entirely know. Normal human cells have built-in control mechanisms. They can divide about 50 times before they self-destruct in a process called apoptosis. This prevents the propagation of genetic errors that creep in after repeated rounds of division. But cancer cells ignore these signals, dividing indefinitely and crowding out normal cells. Still, most cell lines eventually die off, especially outside the human body. Not HeLa though, and that's the part we can't yet explain. Regardless, when Dr. Guy realized he had the first immortal line of human cells, he sent samples to labs all over the world. Soon the world's first cell production facility was churning out 6 trillion HeLa cells a week, and scientists put them to work, in an ethically problematic way, building careers and fortunes off of Henrietta's cells without her or her family's consent, or even knowledge until decades later. The polio epidemic was at its peak in the early 50s. HeLa cells, which easily took up and replicated the virus, allowed Jonas Salk to test his vaccine. They've been used to study diseases including measles, mumps, HIV, and Ebola. We know that human cells have 46 chromosomes because a scientist working with HeLa discovered a chemical that makes chromosomes visible. HeLa cells themselves actually have around 80 highly mutated chromosomes. HeLa cells were the first to be cloned. They've traveled to outer space. Telomerase, an enzyme that helps cancer cells evade destruction by repairing their DNA, was discovered first in HeLa cells. In an interesting turn of fate, Thanks to HeLa, we know that cervical cancer can be caused by a virus called HPV, and now there's a vaccine. HeLa-fueled discoveries have filled thousands of scientific papers, and that number is probably even higher than anyone knows. HeLa cells are so resilient that they can travel on almost any surface. A lab worker's hand, a piece of dust, invading cultures of other cells and taking over like weeds. Countless cures, patents, and discoveries, all made thanks to Henrietta Lacks.
All right, so that was our video over Henrietta Lacks. Your assessment or your assignment is going to be over that as well. So if you need to review this section of the video, I'll also link it on Canvas so that you can review this video as well. That might be a good idea, especially after we have learned about the cell cycle. All right, so this is your learning target. So this is what I want to see you be able to do by the end of this Canvas module. I want you to be able to use a model to illustrate the role of the cell cycle and differentiation in producing and maintaining complex organisms. So we're going to get into that a little bit more. We're going to talk about what the cell cycle is. We're going to talk about cell differentiation within this module. All right, so here is where you need to start taking notes. So on a piece of paper, we need to define the cell cycle. So the cell cycle is the series of events that cells go through as they grow and divide. So we kind of talked about this in the video with normal cells and cancer cells. Both types of cells go through the cell cycle. So our normal cells are going to grow, they're going to divide, which allows things like our hair to grow, our nails to grow, our body to repair itself. And then you have cancer cells that also go through the cell cycle, but they go through this cell cycle too often. So they're growing, they're dividing, they're growing, they're dividing over and over and over and over again. So we're just getting big, large clumps of cells, which become tumors. So we need to talk about what happens during the cell cycle. During the cell cycle, the cell will grow, prepares for cell division, and divides to form two daughter cells, each of which begins the cell cycle again. So the whole point of the cell cycle, the entire reason that our body does the cell cycle, is so that we can go from one cell to two identical cells. So that is the entire purpose of it. We want to go from one cell to two identical cells. Those cells will then be able to go from their individual cells to two more cells, and on and on and on. But essentially this is a process where we start with one cell and then we get an identical exact copy of that cell. That is the end goal, an identical exact copy. So now we have two cells that are exactly the same. So we can see from this little GIF here, I'll start when we start over. So we start as one cell right there with the big eyes, it grows, it replicates its DNA, and then it will start to grow some more. It splits in half, and now we have two identical cells. That is the entire point of the cell cycle. Go from one cell to get two identical cells. So now we need to break down the cell cycle into its parts. So the first part of the cell cycle is called interphase. An interphase is the period of growth that occurs between cell division. So cell division is going to be when our cell splits in half, but before the cell can split in half, it has to prepare itself. So it's preparing itself during this interphase phase. And this interphase is split into three separate phases, I guess. The G1, S, and G2 phase. So we have interphase, and now we've split that into three, G1, S, and G2. And we're going to talk about what happens in each of these components of interphase here in a second. Here's kind of a nice little picture of what's happening. So this orange ring around this circle is the entire interphase part. So this entire orange ring is the interphase. And then if we move in, we have this G1 phase. So the G1 phase is in blue. It is a part of interphase. We have the S phase in red. The S phase is also a part of interphase. And then we have this G2 phase in this like light green color. That is the third part of interphase. So interphase is the entire orange ring around it, and it is comprised of G1, S, and G2. And we're going to talk about what happens in each of these stages of interphase. So first we have the G1 phase. The G1 phase is the period of activity in which cells do most of their growing. Cells will increase in size and synthesize new proteins and organelles. So these proteins and organelles are the things that cells need to function. If the cell is going to split in half, it needs to have double those things. So if it has one mitochondria, now it needs two, so that way when it splits in half, each of those two cells at the end can get a mitochondria. So it's going to do that for all of the contents of the cell. Proteins and organelles are going to double, so that way at the end, when the cell splits in half, it can both of the cells can be identical and have what they need. It's also going to grow a lot because if it didn't grow at all, then the cells would continue to get smaller and smaller as it divided. So it needs to grow to half of its size, so that way each cell can be the same size. So this is what's kind of happening in our G1 phase. Next, if you're still confused about what organelles are, you can write down this definition. So organelles are kind of like the organs of our body, except they're found in our cells. So each one has a different function, and all of the organelles are going to help the cell to survive, essentially. So our next thing is our S phase. And I apologize that there's writing over this, but the S phase replicates chromosomes and synthesizes DNA molecules. When DNA replication is completed, the cell enters the G2 phase. So essentially this is when our DNA is getting replicated. And if you remember, or if you took biology a while ago, DNA is what makes you you. It's what says that you're going to have brown hair, what says you're going to be however tall you are. It says that you're going to need lots of sleep or a little bit of sleep, or you're going to break down food really quickly, or you're not going to break down food quickly. It says what color your skin is, what color your eyes are, how quickly your hair and your nails grow. All of that stuff is determined by DNA. So when a cell splits, every cell has DNA in our body. We have to replicate that DNA. We want to make sure that each cell has the exact same amount of DNA. And in order to do that, if we start out with 23 chromosomes, which is what we have to replicate that. So we have to double it, 46 chromosomes. We have to replicate that. We have to double that number, so that way when the cell splits at the end, we will get the exact same 46 in each cell. So it'll duplicate to 92. 92 divided by 2 is 46. So when the cell splits into 2, each cell will have the exact same amount of DNA. So this is a pretty important step in the cell cycle. Then we have our G2 phase. During G2 phase, many of the organelles and molecules required for cell division are produced. When G2 is completed, the cell is ready to enter the M phase. So this essentially is just kind of more growing, more prepping for that cell to divide. So it's very similar to our G1 phase. We just need to make sure that we have double of everything in our cell, so that way when the cell splits, each cell will have one of everything that it needs. So we just finished talking about our interphase. We just finished talking about G1, S, and G2. I apologize if I was going too fast. You can always go back and rewatch the video. Please also feel free to pause at any point in time. So if you're trying to write stuff down and I'm going too fast, just pause the video, write it down, and then you can keep going. But we just finished our interphase, which is this orange ring. It contains G1, S, and G2. Now we'll get into this very, very tiny M phase. So at this point, we still have one cell. It's a very large cell, and it has double everything that it needs, but we still are considered one cell. So the M phase, or mitosis, is the phase of the cell cycle in which the DNA lines up in the cell so it can split correctly. So everything's going to kind of get pushed off to the side, everything's going to get split up so that there's one of everything on each side, and then eventually our cell will break into two, and we will have two identical cells with one of everything that it needs. So that's that very tiny part of that circle where it says M phase. It's not a part of interphase, it's its own phase called mitosis, or M phase. So now we need to talk about, why do we do the cell cycle? What is the point of the cell cycle? Why do we need to go from one cell to two identical cells? So the first kind of big reason our body goes through the cell cycle is so that new cells can allow organisms to grow in body size by adding new cells. So we obviously all start out as a baby, and we have grown to being a lot larger than we were when we were a baby. So one of the reasons why we're allowed to do that is because our body produces more and more cells, which allows us to physically get larger in size. Same thing with plants, same things with animals, same thing with reptiles. All of those things are going to be going through the cell cycle. Every single living thing goes through the cell cycle in order to grow. larger. Another reason we go through the cell cycle, or our next purpose of the cell cycle, is that new cells replace old cells after they die, allowing organisms to stay alive. So your cells do not stay there forever. So the cells that you had as a baby are no longer the cells that you have now. Because just like us, eventually cells will die, but the cells are able to replace each other. So if I have a cell that dies off, if I have a skin cell that dies off, let's say skin cells die off very quickly, I will have more skin cells to replace that cell, so that way I don't just run out of skin one day. And then the last reason that we go through the cell cycle is that new cells replace damaged or infected cells and tissues, allowing the organism to function correctly. So when you get sick, if you've ever broken a bone or gotten a scrape, gotten a cut, those cells will get damaged. So if your body did not go through the cell cycle, your body would just stay injured forever. So you'd get a cut and you just never heal from it. But because your body goes through the cell cycle, it can replace the damaged and dead skin cells with new healthy skin cells, allowing you to survive and allowing you to stay healthy. So that is the end of this PowerPoint. You need to take the practice assessment on Canvas. Again, it is about the cell cycle as well as the Henrietta Lacks video. So if you do not remember that video, if you need a refresher, please watch that video over again. And then you can start on the next and last set of notes, which will be about cell differentiation.